It's a common assumption that a ship at the bottom of the deep ocean enters a state of permanent suspension. The reasoning is straightforward. Without sunlight or warmth, and under thousands of pounds of water pressure, biological life shouldn't be able to survive, let alone thrive. The RMS Titanic became the ultimate test case for this theory. When it sank in 1912, it settled nearly 4 kilometers below the surface of the Atlantic, deep within a zone previously thought to be biologically dormant. When the wreck was finally located in 1985, researchers expected to find a hull largely protected by the cold and dark. Instead, they found the steel was undergoing a rapid physical transformation. The ship was covered in these porous, icicle-like structures called rusticles. These aren't just simple rust. They are complex formations of corrosive byproducts that indicate the ship is being systematically dismantled. Far from being a stable time capsule, the deep ocean is an aggressive environment that is actively destroying the wreck. This isn't the slow, predictable oxidation typical of saltwater corrosion. The steel is breaking down at a rate that standard chemistry cannot explain. Samples taken from the site revealed that biological activity, not just the water itself, is driving this speed. Current projections show the ship's mass following this curve. Because of this accelerated biological decay, the Titanic is expected to lose its structural integrity and essentially disappear by the year 2030. Understanding how 50,000 tons of steel can vanish so quickly required looking past the hull and into the microscopic details of the rusticles themselves. A piece of maritime history is now on a fixed timeline, being consumed by a force that works 24 hours a day. Researchers analyzing the Titanic's rusticles isolated a unique species of extremophile bacteria, Halomonas titanicae. To survive the pressure and cold, these bacteria use a compound called ectoin. It acts as a stabilizer, allowing their cellular machinery to function in conditions that would crush most other life forms. This is the mechanism of biocorrosion. These microbes use the ship's iron as a direct energy source, cycling carbon, sulfur, and nitrogen as they metabolize the steel. The byproduct of this process is the very corrosion that is weakening the hull. This isn't limited to the deep sea. The Pappy Lane shipwreck rests in the shallow, sunlit waters of North Carolina's Pamlico Sound, yet it shows the same patterns of decay. In this environment, scientists identified a different strain, Mari profundus ferrooxidans 01, performing the same iron-eating role as its deep-sea relatives. These iron-eating bacteria are a universal presence in the ocean, capable of compromising steel structures regardless of depth or temperature. These bacteria are part of a massive ecosystem. A single wreck can host up to 4,800 different microbial species living in a single coordinated community. This is niche partitioning. The ship becomes a structured micrometropolis, zoned by oxygen levels, iron, and leftover fuel. Species settle into specific neighborhoods, much like fungi on a fallen tree. These communities collaborate, cycling nutrients between zones to accelerate the ship's decomposition. The wreck is no longer just a human artifact. Now it has been fully integrated into the local ecosystem as a living reef, designed to recycle its own foundation. This level of environmental specificity makes traditional conservation nearly impossible. A strategy that works for one wreck may be completely ineffective on another, because the underlying microbial communities are so different. Preserving these sites requires developing site-specific detection and mitigation strategies that account for the unique biology of each individual ship. For international organizations like UNESCO, this shifts the challenge from general site management to the need for continuous, microscopic monitoring of thousands of different locations. Protecting our history means finding ways to stop a microscopic process that has had millions of years to perfect its efficiency. While these biological processes are a problem for historians, they are becoming a focus for environmental engineering. Researchers are looking at these iron-eating microbes as a potential tool for bioremediation, using natural biological processes to address human-made pollution. The same metabolic pathways that Halomonas titanica uses to consume the Titanic can be directed toward industrial metal waste and toxic runoff, breaking down pollutants that are otherwise difficult to remove from the ocean. The biological mechanisms currently dismantling maritime history provide the exact template needed to clean the oceans of the future.